Hello YouTube, Joseph here with Prep for Step, and in this lecture I'm going to discuss the cyclic AMP or the cAMP signaling pathway. It is a secondary messaging pathway that is used by a lot of endocrine hormones to cause an effect in their target cells. So it's pretty important to actually know, but it's very easy to understand. And so let's get started. It's pretty brief. I'll go over it as quick as possible. Okay, so the first thing we need to know, like we always do, we always try to find out what is even cyclic AMP? What does that mean? Well, most of us know what adenosine monophosphate is, right? Adenosine monophosphate is just an adenosine with a sugar, and over here we have the phosphate group. If we attach two more phosphate groups to this, it makes adenosine triphosphate, or our source of energy, which is the energy is basically in these bonds, right? So that's ATP, that's AMP. But what is cyclic AMP? Cyclic AMP, the only difference between AMP and cyclic AMP is this ring right here, okay? There is a ring over here that you don't see it on that side, right? So this ring makes the difference between cyclic AMP, and there's an enzyme that actually does this, and that enzyme is called adenyl cyclase, which we'll discuss in a bit. Okay, so let's see what type of hormones actually use the signaling pathway. And this is pretty good for the, the guys that are actually studying for step one. The first one is going to be the mnemonic used to memorize the names is called flat cAMP, right? And each of these stands for something. The first one is FSH. The L is LH. The A is ACTH. The T is TSH. The C is CRH. The little h stands for HCG. The A stands for ADH, but ADH, also called vasopressin, has two different types of receptors. One is V1 and the other receptor is V2. The V1 actually is vasopressin type 1, which actually works in the vessels, causing pressing of the vessels, hence the name vasopressin. The V2 actually works in the kidneys, and it causes a function actually in the kidneys. So this is the one that we're actually talking about. The V1 does not work with cAMP. The M is going to stand for MSH, or melanocyte stimulating hormone, and the P is actually PTH. Okay, so that's the mnemonic. There are a few other ones that actually use this. One is GHRH, one is calcitonin, and one is glucagon. Okay, so that's pretty much what covers all the endocrine hormones that actually use this pathway. Okay, so now let's bring all the information that we have together and see how the system works all together when put in one picture. Well, first off, we have to have the hormone or the ligand or something that's going to stimulate this receptor. It all starts from right here. Now, the endocrine hormones we already talked about, the LH, the flat cAMP, basically, all of those can actually work on this receptor. And every single one will have a different receptor. Now, these receptors can either be bound to a G protein that is stimulatory, or it can be inhibitory, which I'll tell you exactly what that means in a second. Okay, so any of those endocrine hormones that we talked about can stimulate a receptor. This receptor, in turn, by itself, it's bound to a transmembrane protein. Okay, this transmembrane protein, once activated, is going to stimulate the G protein. This right here is the G protein that we're talking about. It is a trimeric protein, means it has three subunits, all right, alpha, beta, and gamma. And it can either be a stimulatory, GS, or it could be an inhibitory, which is GI. And that's what is going to determine that is the receptor over here. One of the receptors that we know is bound to, most of the receptors are bound to GS, okay? Most of the receptors are going to be stimulatory. They're bound to a GS, or they're called GS coupled. But there's one particular one that you really need to know. It's alpha-2 receptor. If this was an alpha-2 receptor for epinephrine, that means that epinephrine would come to work on this receptor, and this would be an alpha-2. That would be bound to an GI. That's the only one that really is significant for us to know. The rest of the ones, all the flat cAMP, are going to be stimulatory. So once this protein, this G protein, this trimeric G protein is stimulated, in turn it's going to stimulate something else. This enzyme is called adenyl cyclase. Now, we previously talked about this just for very briefly, but if you had to take a wild guess, what do you think adenyl cyclase is going to do? Well, it's going to cyclase something. It's going to make something cyclic, right? And what it's going to do is going to take ATP and turn ATP into a cyclic AMP. So this is where the name comes from, cyclic AMP. And we know that the difference between ATP and cyclic AMP is pretty much lose 2 phosphates and take the last phosphate group and convert it into a little cycle or a circle. And that's pretty much what it does. The cyclic AMP in turn is going to stimulate another enzyme. This enzyme is very important. Okay, so this enzyme is called pKa, protein kinase A. Don't worry about it, but PK, protein kinase. So what do you think this is going to do? Previously, we've discussed this in other videos that anything that's kinase is going to take ATP and it's going to give out AMP, and it's going to take those phosphates and it's going to phosphorylate different proteins. Okay, so if I had an enzyme here, what do you think I'm going to have over here if it was stimulated by pKa? Well, the enzyme would get phosphorylated. So pKa, protein kinase A, will phosphorylate proteins and enzymes inside the cytoplasm. But it also does something else, and that's actually the important part. And a lot of questions on step 1, if they were to ask this, would be about this particular part. Not only does protein kinase A phosphorylate some enzymes here, but it also phosphorylates something very important, and that is called the CREB. Now, what is CREB? Well, it is cyclic AMP, responsive element binding protein. Okay, some people put a P here, some people don't, right? So pKa phosphorylates this protein. Once this CREB, or this cAMP response element binding protein, is phosphorylated, it activates. Then it diffuses inside the cell, inside the nucleus, and it actually works on this particular thing that you see right here, CRE, which is the cAMP responsive element. It's basically a promoter on the DNA that causes transcription of particular genes. So one last time, cAMP is increased, stimulates pKa. pKa is going to phosphorylate some enzymes, right? And that's what you get right here, phosphorylated enzymes. The next thing it's going to do is going to phosphorylate a particular transcription factor. This CREB is a transcription factor. CREB is cAMP response element binding protein. It phosphorylates it. It diffuses across, this CREB diffuses across into the nucleus, or comes across into the nucleus, binds to this CRE, or cAMP response element, which is a portion or promoter on the DNA. Once this is activated, it's going to start promoting the transcription of a particular sequence or a particular gene. And then you get the result, which would be some kind of protein or some kind of enzyme. Okay, I hope this helps. Please like, share, and subscribe, and we'll see you guys next time.